Hello everyone and welcome to the Science AAAS webinar, Deciphering Cancer, Genomic Instability in Cancer. I'm Sean Sanders, Editor for Custom Publishing at Science, and I'll be moderator for this webinar event. While it is well established that DNA damage can increase the risk of cancer, changes to the epigenome or the chromatin architecture are equally important. DNA damage triggers a redistribution of DNA binding proteins around the site of damage, resulting in localized and temporary alteration of chromatin structure. However, repeated cycles of DNA damage and repair may lead to permanent changes in the epigenome, thereby promoting the onset of diseases such as cancer. This webinar will examine how we may be able to develop effective new therapeutic options for cancer treatment by targeting the proteins responsible for chromatin modifications. We will discuss the different forms of DNA damage that lead to genomic instability and tumorigenesis, including copy number gains, and how understanding genomic instability can inform the development of cancer treatments. I'm very pleased to introduce our speakers for today to you now. They are Dr. Johnathan Whetstine from Massachusetts General Hospital and Harvard Medical School in Charlestown, Massachusetts, and Dr. Mark O'Connor from AstraZeneca in Cambridge in the UK. Thank you both so much for joining us today. Before we get started, I'd like to share some information for our online viewers. At the top right of your screen, you will find photographs of today's speakers and a View Bio link, which you can click on to read more details about their background and research. Underneath the slide viewer is the Resources tab, where you can find additional information about technologies related to today's discussion. After the speaker presentations, we will have a short Q&A session, during which we will address some of the questions submitted by our live online viewers. So if you are joining us live, start thinking about some questions now and submit them at any time by clicking the Ask a Question button below the slide window, typing the question into the message box, and clicking OK. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the top left of the screen. For tweets, you can add the hashtag #ScienceWebinar. Finally, thank you to Cell Signaling Technology for sponsoring today's webinar. Now, I'd like to introduce our first speaker, Dr. Johnathan Whetstine. Dr. Whetstine is currently Associate Professor at Massachusetts General Hospital and Harvard Medical School. His laboratory has established that chromatin factors are capable of driving DNA copy number alterations at specific sites within the genome that are associated with drug resistance. This discovery changes how we perceive the regulation of DNA replication and establishes new biomarkers and therapeutic targets for reducing the selection of drug-resistant DNA changes. Dr. Whetstine's lab is examining the fundamental role of genotypes play in cancer research, which may ultimately lead to personalized treatments, resulting in better outcomes. A warm welcome to you, Dr. Whetstine, and thank you so much for being on the line.
It's my pleasure. I want to really thank Science for putting together this special webinar. The work that I'm going to tell you about today is centered around how epigenetics or chromatin factors are able to facilitate copy number amplification at distinct regions in the genome, especially those that are affiliated with drug response or drug resistance. The picture that's on the slide actually is kind of the picture that captures all the entire story in many ways. You can think of this as there's a factor that's able to obtain or results in a site-specific re-replication and amplification event. So my lab is interested in understanding how and why cancer cells quit responding. Our ultimate goal is to better understand and develop better treatment options, be that through epigenetic mechanisms or through targeting those processes, or thinking about how we would make predictions about why an individual that responds ultimately quits failing, and thinking about new biomarkers or new tools. And how we're going to get at this is we're very interested in looking at it from a mechanistic point of view, and by understanding kind of the details. And we believe that epigenetics or chromatin modulation is an essential piece to this equation of resistance, and also becomes a really critical hallmark for us to think about in developing biomarkers and therapeutic targets. So what is the big question that where we think this plays a major role? We believe this plays a key role in the context of tumor heterogeneity. We know that tumor heterogeneity is a major clinical problem. We know that you can have two adjacent cells within a field, and one can have, parental copies, a mom-dad allele, as seen with the red and the green of 2 and 2, but it can have adjacent cells that have 3 copies in one allele or 4 copies in another allele. We know that from all the mutagenic data and all of the analysis of tumors that it's very unlikely that there's that much robust mutation and alteration happening in each division. And so what we become curious about is understanding, are there plastic responses, or how is the genome potentially being regulated in a very systematic way that would allow these to present themselves and then kind of dissipate. One might ask, why do you care? Well, it comes down to amplifications or changes are linked to drug resistance and poor prognosis. And so, in fact, many examples exist in the literature, and I'm going to show this particular example because it's relevant for the story that I'm going to tell you about, is that there's certain amplifications and copy gains that are seen. 1q21, that locus between 1q12 and 1q21 on chromosome 1, that region has been seen in up to 25% of tumors. Matt Myerson's group in 2010 made this observation, and in that there was the assumption post that discovery that there would be a critical oncogene or a critical pro-survival factor that would dictate the consequence. And it turns out that as we've looked at tumors, and the field has studied this, they found that there's actually a consistent cytogenetic relationship, but the use of the oncogene or pro-survival factor has changed, and there's a list of various ones on the top. And this has been linked to, once again, resistance and poor prognosis. So what I would like to do is, as we begin this, I want you to think of these cytogenetic changes as more like a DNA suitcase, in the sense that within that cytogenetic DNA, it carries a systematic collection of genes that, depending on the cell, will determine potentially which oncogene or pro-survival factor it will use. And as you go through the talk, you'll see why we believe this is probably what's happening. But it's also really important to realize that in the context of amplifications, that they are not always permanent. In fact, I think with the advent of all the digital sequencing, a lot of people have made some very strong assumptions that all the genetic changes they see, especially in the context of amplification or copy gains of regions or genes, that they have to be permanently acquired in the genome. And this is not true because this has been dating back to the late 50s until now, where we know that, for example, upon methotrexate therapy, dihydrofolate reductase, indicated here in red, will become amplified, and it can present itself 2 ways. And in fact, it has presented itself 2 ways in both patients and in culture. That is that it's integrated in the context of an HSR, or in double minutes with these extra chromosomal fragments or components that are not perfectly inherited. But then there's also another really good example of this, which really hallmarks and highlights the plasticity of the system, is the EGFR. EGFR amplification, upon treatment, will result in resistance because the amplifications disappear. Interestingly enough, upon removal of the drug, these will reappear again in the samples or in the tumor. This really, for us, was a key set of observations because this begins to tell us that there's some plasticity in the context of DNA amplification. And since we know that the epigenome itself is very plastic, this raises the possibility that the epigenetic state or the chromatin states themselves are critical features in how you would potentially promote amplification of distinct regions in the genome. What I'm going to do is I'm going to tell you about how we discovered the factor, but first we'll kind of highlight what kind of enzyme it is. In fact, it's a Jumanji demethylase. Jumanji enzymes require cofactors, oxygen and iron, as well as alpha-ketoglutarate to execute their function. It's also important to realize that there's 2 important points here. There is a byproduct, succinate. Succinate is a natural inhibitor, and we'll show you how that can feed back into this system, giving you a way to shut down the enzyme and potentially could contribute to the observed heterogeneity. The second part is oxygen. The field has assumed for many years that oxygen is in fact it's positive, and when you get reduced oxygen, it would shut these enzymes down. And the story I'm going to show you in the data I'll show you is actually showing that's not the case at all. In fact, you get a very different effect. With reduced oxygen levels, especially physiologic levels, you actually find that the protein becomes very, very stable. So which Jumanjis am I going to tell you about? The Jumanjis that we're going to discuss today are the KDM4 or JMJD2 tri-demethylases. They've been implicated in a wide range of events. We've shown their roles in replication timing. We've shown they can cause replicative stress. Others have shown they play roles in transcription and gene control and damage response and various other systems. The specific family member that we'll tell you about, or I'm going to tell you about today, is the KDM4A enzyme, or JNJD2A. It had a C. elegans homolog, which allowed us to actually associate its function with DNA replication. So what do we know about this role of this enzyme? We know that it is able to physically generate, through the demethylation of K9 and 36, methylation, which are its substrates, it's able to go from promoting 2 copies to 3 copies of a very distinct locus, the 1Q21, 1Q12 to 21 region. We know that overexpression causes this. We term this event transient site-specific copy gains, or TSSGs, and I'm going to begin to highlight what that actually means in the context of amplification. What we ended up demonstrating was that this requires catalytic activity, which opens up the idea or the possibility that therapeutic intervention may be something that's important, and I'll come back to that in a few minutes. We demonstrated that the tutor domains, which are the reading modules that help recruit the enzyme, are essential, indicating that there's an innate ability or something that helps target this enzyme, and that's part of the function. Tutor domains also pose a unique opportunity for therapeutic intervention in the future. We further demonstrated in collaboration with Dave Alice's group that if you introduce into cells a lysine to methionine substitution at the 2 sites that are its substrates, in fact, they will cause site-specific copy gain as well. We altered other residues on the histone tail and they didn't do it. So what this begins to tell us is that the chromatin, the epigenetic state, is actually essential. We didn't manipulate the enzyme in that context, but in fact, their substrate. So this documents for the first time that the epigenetic state or chromatin states at distinct loci will give a region of predilection to undergo DNA amplification. So what we ended up doing, and this becomes very important, is all our original data was generated in cell models. But what then we've done is we've built, using TCGA data in collaboration with Getty Getz' laboratory, an informatics way to basically take thousands of tumors, exomes, RNA-seq, etc., combine that with either gene signatures or epigenetic factors or oncogenes of choice. We're able to generate systematic, what we call cytoplots, which will evaluate whether copy gains or gene expression association. And then what we're able to do then is, based on these predictive models, go back to tissue culture. So based on using this type of strategy, this is how we were able to identify the regions that we found that were downstream of the demethylase in the context of tumors. And here's just kind of an illustration of that data. Each dot represents a cytoplot, or a cytoband. And what you can see, the blue line indicates the amplified site for KDM4A in tumors. We found that KDM4A is amplified in approximately 19% of tumors that we've evaluated. And that, at that time, it was around 4,000 tumors and 12 tumor types. Since then, we've expanded it to more than 19,000 tumors and more than 23 tumor types and seen comparable data. What you see there in the light blue shaded region is, in fact, that 1Q12 to 21 region. And so we had seen in the lab that it altered these in cells, but then we wanted to digitally see if this would make predictions. And so it had, in fact, predicted those regions, and then using the statistical cutoff of where the values are, we went back to the lab, and in every cell type that we've been able to evaluate, we can recapitulate the data. What we find is where the stats are for the cytobands, the copy gains occur, and you can see with 1Q12H, 1Q1221, 1Q21.2 here. But at 23.3, we see no change. And you can see that we can recapitulate that in the cells. And we've now done this in numerous cell types. I'll show you data we've done at primary cells. What's interesting, though, is we know that from cytogenetics or using cytology such as spectrokaryotyping and G-band, there's no global defects in chromosomes or their regulation. Consistent with this, what we find is that in many cases, these cytogenetic changes or these loci that are becoming copy gained or amplified are in some cases mutually exclusive, suggesting that they're actually small components, small extrachromosomal parts, which we were able to demonstrate by coupling interface fish with DNA fish. So from there, what we've gone on to do is we see that we can get these effects of site specificity. We went on and demonstrated with this, though, that there was a key feature to this that was kind of unprecedented and began to start to show us how the epigenetics or chromatin changes could influence tumor heterogeneity for copy number. What we found is that they required S phase. S phase being that this is when DNA is being replicated. This is when the cell is copying so that it gets the two daughter cells. And if cells are not copying or they're not replicating or they're not in S phase, we do not see the copy gains. So what does that mean? What does that really look like? Well, here's an example that we've done where we take cells that are stably expressing or overexpressing the demethylase Kadium-4A, mirroring that of what we see in a tumor overexpressor. And what you can see is that if we arrest it, which is the 0 hours in hydroxyurea, which puts them right at the G1S boundary line, if you release them into S phase, you'll start to see copy gains occur at the sites that are downstream of Kadium-4A. But what's really remarkable, and I think extremely important, and a very major focus for us in the future as we're moving forward here, is that we notice the copy gains are removed. They disappear as you go to late G2. We know that there's different kinetics for the different regions, suggesting there's an enzymatic or a possible role there. And more consistently and published in something we've seen through a small screen we conducted is if we actually screen a number of molecules, and you can see the one that says 10, 11, in fact, it gives the G1S a rest, but what we actually see is you get retention of the copy gain. This begins to tell us that there is a molecular target, something that we can use a small molecule to interfere with, consistent with the data that I just showed you above, and consistent with the fact that different regions that amplify have different kinetics of removal. And this is something that we're actively pursuing and predict is probably highly altered in tumors, especially tumors that present a lot of genomic instability. From there, what we've gone on to do is ask a slightly different approach. What we found was relevant to tumors, and is that a pathology in tumors have come up with a mechanism to amplify and generate regions that are filled with these pro-survival factors, or is there a biology that's in place that allows this to be actively selected for, and then what cancer's done is just hijacked it. So it's not that it's smarter than the average cell, it just uses a process. And so what we did is we screened a number of physiological triggers for this, and what I'm going to tell you about is what we've actually found from this study. What we actually found is that, in fact, hypoxia, in fact, is a major driver of these events. We find that, in fact, we see this in tumor cells. We see this in primary cells indicated here. What we've been able to do is we treat cells with normoxia or hypoxia. We give them cytokines to either proliferate or not. And what you can see on the far right side of the graph there, you can see that with 1% oxygen, which is a physiologic hypoxia, plus stimulation, you get copy gain very selectively. You don't change the whole genome. We did evaluations, of course, to look at that. We found the genome was fairly stable. More importantly, we used our digital analysis, and the data is quite striking. If you use a hypoxic signature, gene signature, you can see those that present the signature in red, and this is showing breast cancer. We've done the exact same data in over 1,000 lung cancer samples as well. You get those cytogenetic changes where we see them in culture. More importantly, if you look at tumors that don't carry a signature, they don't have them, indicated by the light blue region. So what was quite amazing about this is it really illustrates how the process is actually a physiologic process and being regulated. So we ended up demonstrating through that this was through Kadium-4A stabilization, that hypoxia stabilized the enzyme and allowed it to function, and it allowed it to execute the process. We further demonstrated with KDM inhibitors that we could shut that process down. So this then brought us to ask another question. Are there other epigenetic factors, other processes that could crosstalk to these events, especially those regulating the 1Q21 transient site-specific copy gain? And what we've been able to demonstrate in a more recent study was that microRNAs on miR23A and miR23B as miR137, they repress Kadium-4A. And in fact, if you put those mirror mimics in cells that are hypoxic, you can shut off copy gain. They're able to suppress it. We also did the inverse, where we put anti-mirrors against them. And what you can see here in the two graphs, the upper one demonstrates you get increased copy number of CKS1B, which is in fact a very important pro-survival oncogene associated with myeloma and breast cancer and a few other cancer types. You see that it, in fact, is copy gain, and more importantly, you see that there is a transcriptional dosage increase. So this prompted us to ask a different question. These mirrors happen to coincide with different regions, some on different chromosomes. So what we wanted to do is see if we looked in tumors using our digital approach, does it make any additional prediction? Does it hold up? And what we actually found was that if you look in the tumors, that when you get a microRNA deletion indicated with that kind of stop sign there over the microRNA, we did get, in fact, increases in 4A. We saw increases in CTS1B transcripts and copy gains in the tumors. So what we were able to demonstrate is that you can have KDM4 amplification, which will drive copy gain. We have hypoxic exposure indicated with the reduced oxygen level there, which will drive it. We can have microRNA deletions on different chromosomes that are regulating it and in turn cause other regions to be amplified. And we also know that metabolic states can be very important, which I didn't show you. But what we went on to do is show that through microRNAs and KDM4 related or KDM inhibitors that these are in fact druggable. And now what we're doing is based on these results, we're expanding this out to identify other additional epigenetic mechanisms that may be influencing amplifications in the genome in site-selective ways. We're looking for other metabolites or growth factors that could also be stimulating these events, which may begin to help us understand how heterogeneity can happen. And so what does this mean in a global picture in the context of a clinical context? It's really important to realize that what we demonstrated is an amplification, overexpression, or hypoxia can drive this through 4A. And we can get an amplification event of 1Q21. We get these regions, which we'd almost consider as DNA suitcases. We then went on to demonstrate, which is something that's quite important, is that if you get a deletion, say in the context of the microRNA on chromosome 19, this in fact can also promote copy gains of a different chromosome, suggesting that some of these events that we see by looking at the cancer genome data, they actually may be linked and associated. And so what we're now doing is we're asking a separate question: does this potentially shed light onto the original data about you get a consistent amplification but not the same gene? And the answer looks like yes in our data that we're doing across the panel of cell types. Epithelial cells, you see that these pro-survival oncogenes that have been associated with various cancers, they don't change. You'll see one selected in breast, but you'll see both in myeloma. And so we believe that the neck of the hydra is really the copy amplification. And therefore what we're going to do, and we're doing, is looking to see if these effects of KDM4 factors are in fact associated with a late-stage disease. This also opens up a unique window to start thinking about therapeutic intervention to help prevent resistant selection and potentially extend that survival. So in closing, basically what we've shown is that epigenetic factors and chromatin factors can in fact regulate site specificity. And the key now for the field is to really identify regulatory pathways and other factors and build a compendium of those so we know what would be good targets or biomarkers. And with that, I have to thank the group. It's a fantastic group to work with. Our funding sources, because without money you can't make anything happen. And of course, collaborators allow us to ask very diverse sets of questions. And of course, there are positions in the lab. Thank you.
Wonderful. Thank you so much, Dr. Whetstine. And we're going to move right along to our second speaker. And that today is Dr. Mark O'Connor. Dr. O'Connor is Chief Scientist in Oncology at AstraZeneca, based in Cambridge in the United Kingdom, where he heads up the DNA damage response area. He has provided scientific leadership on Olaparib, a first-in-class oral PARP inhibitor and the first cancer medicine based on a targeted inhibitor of the DNA damage response. Dr. O'Connor has overseen the growth of DDR within AstraZeneca to an industry-leading portfolio comprising 7 DDR projects with 5 inhibitors currently in clinical development. Thank you so much for being on the line, Dr. O'Connor, and over to you.
Thank you very much, Sean, and thanks to Science for this opportunity to talk to you today about targeting the DNA damage response and genomic and epigenomic instability to generate new cancer therapies. So the DNA damage response, or DDR, is actually a broad term for the many different responses to DNA damage. So it's not only the response to damage that leads to cell cycle arrest and the repair of that damage, but actually also is involved in decisions on cell death or senescence. There are multiple different interactions with the immune response. And importantly, one of the sort of critical aspects of the DNA damage response is the regulation of chromatin structure and also gene expression. So chromatin regulation has 2 major factors associated with that when it comes to DNA damage response. The first is the need to open up the chromatin to allow access to the repair factors. And the second is that many DDR factors also are cofactors for gene expression and transcription regulation that in turn provide further products to enable the DNA damage response to occur effectively. So the multiple DNA damage response pathways have evolved to deal with the many different forms of DNA damage. What I'm going to focus on today are really the two major forms of damage. First of all, single-strand breaks, which are thought to occur in the tens of thousands in every cell every day, and also the most cytotoxic lesion, the DNA double-strand break. So the poly-ADP ribose polymerase, or PARP protein, is one of the key proteins that is involved in the induction of repair of the single-strand breaks. In terms of dealing with DNA double-strand breaks, there are two main pathways. One pathway, the non-homologous end-joining pathway, which can occur at any point within the cell cycle, is regulated by the DNA-dependent protein kinase, or DNAPK. But that tends to be more error-prone. And if possible, what cells will do is they'll use the undamaged sister chromatid following replication and use homologous recombination repair, one of the key factors of which is ATR. And the ATM protein is involved in the processing of DNA to actually facilitate both of those forms of DNA double-strand break repair. So ATR, ATM, and DNAPK are all kinases part of the PR3 kinase, like kinase family. But ATR, ATM, DNAPK, and PARP, as well as being involved primarily in repair, they're also involved in modifying chromatin. In addition to the actual repair pathways and the roles of ATM and ATR in the repair process itself, they also play key roles in cell cycle checkpoint regulation. And in fact, there are additional proteins that we know are key to being able to provide time to repair the DNA damage. So, for example, in G1S checkpoint, ATM, along with CHEK2 and P53, are key G1S checkpoint players and also potentially targets for cancer therapies. In S phase, where it's really important to provide an opportunity to repair damage before the replication fork reaches it, or if there are issues with replication fork stalling and collapse, then again you have key players such as ATR, CHEK1, DNAPK, and WEE1 kinase that are involved in that aspect. And then finally, the G2M checkpoint is really one of the last opportunities to deal with DNA double-strand breaks ahead of cell division, although we're now also learning that there are DDR aspects in mitosis as well that can also be really important to prevent mitotic catastrophe. So given that the DNA damage response is so germane to cells, why do we think we can target the DNA damage response in cancer? And really the answer comes down to the fact that there are at least three major aspects of DDR that are different in cancer cells. So the first, which has really been the basis of classical treatment for cancer, so radiotherapy and DNA damaging chemotherapy, is based on the fact that there are increased levels of endogenous DNA damage and greater levels of genomic instability that make cancers more sensitive to DNA damaging agents than normal cells. But what I'm going to focus on today are two other aspects. So one is the fact that cancers have increased levels of replication stress, and that the replication stress response that involves ATR, CHEK, DNAPK, and WEE1 proteins actually provides a really good opportunity to specifically target cancer cells. And the second is the fact that one of the very early events in the formation of a cancer is the loss of one or more DDR pathway proteins. And this in turn results in a greater dependency on the remaining DDR pathways. And the next slide really provides that schema of how we think the loss of a DDR protein or pathway occurs during cancer formation. So what we know is that while there's not redundancy as such, many different DDR proteins can complement for one another. They can compensate in the absence of activity. So in a normal cell, though, there might be a number of different DDR proteins or pathways that can deal with a particular type of damage. In this case, we're representing that by two pathways, A and B. What happens in precancerous cell is that oncogenic drivers lead to increased endogenous DNA damage and the activation of one or more pathways. So that's represented by the activation of pathway A in this case. But that's almost like having the accelerator on and the brake at the same time in terms of having the oncogenic driver, but also the cell cycle checkpoint activation. And if a cancer forms, what will have invariably happened is a selection for the loss of that DDR pathway. So in this case, we lose pathway A in the cancer cell specifically, and that leads to a greater dependency on the remaining pathway, pathway B. So the idea there is that we have an opportunity to come in with an inhibitor of pathway B and therefore induce cancer-specific cell death in the process that's been called synthetic lethality, whereas normal cells will still have their backup pathways to compensate. And the one really great example of synthetic lethality is the sensitivity of cells that have lost BRCA gene function, the BRCA genes being involved in homologous recombination repair, double-strand break repair, and that have been linked to increased risk of both breast cancer and ovarian cancer, as well as prostate cancer in men. And in 2005, two back-to-back papers in Nature demonstrated this amazing level of sensitivity of BRCA-deficient cells to PARP inhibitors. In this particular case, what you can see in the figure on the right is that the BRCA-deficient cells are more than 1,000-fold more sensitive to PARP inhibitors than wild-type cells, or even cells that are heterozygous for BRCA2. Now that's really important because in patients who've inherited one functionally defunct copy of BRCA, they will still have a functional copy that provides them essentially with protection against PARP inhibitors. But in a tumor cell that has already started with the loss of one BRCA mutant allele, the loss of the second allele makes them very sensitive to PARP inhibitors. So that is an example of germline BRCA deficiency, and it's also possible to lose both copies, of course, somatically in a cancer cell. And it was really the basis of this original finding in 2005 that led to the generation of PARP inhibitors and their use as single agents in BRCA-deficient cancer treatment. The first one I'm going to do is I'm going to talk a little bit about PARP and its role in the single-strand break repair process. So PARP protein is a zinc finger protein that will bind to single-strand breaks, and upon binding it will utilize NAD to form these poly-ADP ribose chains. Now, the ability to form poly-ADP ribose is important not only for chromatin modification and opening up the chromatin to allow the access of repair factors, but importantly, one of the key aspects is PARP automodification. And by modifying itself, that allows the dissociation of PARP from the DNA, and subsequently, it allows the assembly of repair factors onto the single-strand break that can then facilitate the repair of that DNA damage. The poly-ADP ribose chains are then broken down by glycohydrolase so that PARP can then be recycled to repair other single-strand breaks that are occurring in the cell. So what PARP inhibitors do is they bind to the active site of PARP and they prevent PARP from utilizing NAD. And not only does that prevent the repair because of opening up of the chromatin, but also importantly, it traps PARP onto that single-strand break. Now, what's been demonstrated is that the PARP trapping is consistent amongst many of the PARP inhibitors that are currently being investigated in the clinic. And the idea is that the longer the PARP is trapped onto the DNA, the greater the chance that the replication fork will reach that trapped PARP onto the DNA, and you will get a stalling of the replication fork and then cleavage of the DNA, what's called collapsing of the replication fork, resulting in a one-ended DNA double-strand break. Now, in normal cells, those DNA double-strand breaks generated in S phase will be repaired by the homologous recombination repair pathway. But in cancer cells that are deficient in homologous recombination, so HRD, for example, those that have non-functioning BRCA protein, they will end up utilizing the error-prone pathways such as classical non-homologous end joining, which over multiple rounds of replication will lead to the accumulation of genomic instability, and ultimately that genomic instability is no longer consistent with cell survival, and you'll get cancer cell death. So this example really of a PARP inhibitor and its ability to specifically kill BRCA-deficient cancer cells was the first example to be demonstrated in the clinic, and it led to the publication of this New England Journal of Medicine. And in fact, what I'm showing you at the top here is a radiograph of the very first patient who responded to single-agent PARP inhibitor, where this patient had a BRCA1 deficient cancer. And you can see the peritoneal mass shrunk over a number of months following treatment with Olaparib. And following this phase 1 trial, there were then a number of additional clinical trials that demonstrated PARP inhibitors' ability to shrink tumors that had BRCA deficiencies. And indeed, further clinical trials went on to demonstrate in high-grade serous ovarian cancer that there was a broader activity beyond BRCA1 and BRCA2 that was often associated either with epigenetic silencing of BRCA1 or the mutations in other homologous recombination repair genes such as RAD51 and ATM and so on. So in addition to the ability to use PARP inhibitors as monotherapy in patients who have tumors that have BRCA mutations, it was also demonstrated that PARP inhibitor treatment following platinum treatment that effectively shrunk tumors in ovarian cancer patients could maintain that tumor aggression for a longer period of time and provide the patient with an opportunity to be free of chemotherapy treatment. And PARP inhibitor treatment was seen to be much better tolerated because of that specificity and the impact on cancer cells rather than normal cells. And so what's happened in the last few years is that there have been a number of trials and those that have led to the approval of Olaparib in high-grade serous ovarian cancer patients where the tumors are platinum sensitive and are then essentially treated with PARP inhibitors in a maintenance therapy. And high-grade serous ovarian cancer is very different to other types of ovarian cancer in that it's associated with a high level of genomic instability and chromosomal copy number changes. And in addition, it's associated with loss of the homologous recombination repair pathway that also deals with platinum damage. And that's really where the connection comes from. So what I'm showing you here is study 19, clinical trial outline. And essentially what this involved was randomizing patients who responded to their platinum treatment either onto Olaparib, the PARP inhibitor, or onto placebo. And the idea was to follow how long it took before the tumors grew out following that platinum tumor shrinkage. The primary endpoint was something called progression-free survival. And I'm just about to show you the data here. What was demonstrated was that there was activity both in tumors that had BRCA mutations, inherited BRCA mutations, but also in those BRCA wild-type tumors, where we believe that a lot of that activity is driven either by somatic mutations in BRCA or epigenetic silencing of BRCA1, or indeed mutations in other components of homologous recombination pathway. So just for example, what you can see here on this Kaplan-Meier plot is in green, where the patients had tumors with BRCA mutations. You can see that it takes much longer before their tumors progress. So essentially, every time the plot goes down, that's the patient who's progressed, whose tumors progressed. And you can see that it takes much longer for the tumors to progress on Olaparib treatment compared with the placebo arm, which is in the black or the dark blue. And in fact, what we now know is that many of these patients have gone on to respond to Olaparib single agent treatment for many years. So what I'm going to do in the last few minutes is really talk about another aspect of DDR targeting, which is based on this idea that replication stress levels are much higher in cancers compared to normal tissue. And replication stress can be defined in a number of ways, but perhaps one of the more useful ways of describing it is an uncoupling of the DNA polymerase activity from the riposome helicase activity. So as the helicase unwinds the DNA, the polymerase should be coming behind it, and it should be synthesizing the nascent DNA strand. But if you end up having some blockage or delay in the ability of the polymerase to be able to synthesize DNA, then what you end up with is an extended region of single-strand DNA, which is very sensitive to cleavage. There are many different mechanisms by which you can increase replication stress. So, for example, if cells have gone into S phase too early, or they have genetic deficiencies that essentially mean that there is not a sufficient level of DNTPs, then you can have replication stalling. You can have replication fork stalling, and then that will lead to this extended region of single-strand DNA. Other mechanisms by which you can end up having an imbalance between replication and DNTPs is if you have a deregulation in the number of origins that are being fired, and that can occur when you have amplification of cyclin E, for example. You can also end up with increased replication stress with oncogenic proteins such as MYC being overexpressed, and again that's thought to drive transcriptome, and you get clashes between the transcriptome and the replosome. Or indeed you can have DNA-bound obstacles such as trapped PARP or secondary structures, for example, or other DNA lesions. Now what happens if you have that extended single-strand region is that it has to be protected by replication protein A, and what you end up with is a filament of replication protein A that itself then attracts an ATR complex and initiates the replication stress response. Now, replication stress response involves many factors. So it will, first of all, it will involve the replication fork stabilization and the inhibition of endonucleases that otherwise would cleave that replication fork and lead to its collapse. It also involves the control of replication origin firing, as well as the activation of DNTPs through CDK2. Now, what you see here is that ATR will initiate not only its own replication stress response, but will also activate downstream DDR proteins such as CHEK1 and WEE1. What happens if that replication stress response is inhibited is that you will end up getting these single-end double-strand breaks at the replication fork and an activation of the double-strand break repair pathways that have been shown to involve ATM, ATR, and more recently also been shown to involve the role of PARP1. And so one of the questions that we've had really within AstraZeneca is whether or not we can target cancers with high levels of replication stress, potentially because they have genetic deficiencies that increase the likelihood of replication stress, or also the possibility of increasing that replication stress through combinations, for example, with PARP inhibitors such as Olaparib. So you can see that there are multiple points in the replication stress response where it's going to be possible to target those DDR factors and potentially lead to cancer cell death. So one of the aspects that I mentioned was the potential to impact on the levels of DNTPs. And WEE1 regulates DNTPs through its control of CDK2, which in turn controls E2F transcription factor and ribonucleotide reductase M2 subunit, which in turn helps to provide the pool of DNTPs for replication. The consequences of inhibiting WEE1, therefore, are to increase essentially the activity of CDK2 to provide aberrant DNA replication, and also increase activity of CDK1 that normally prevents inappropriate G2M transition. And so really the idea behind a WEE1 inhibitor is that you can have enhanced DNA damage in S phase occurring in cancer cells, either because they're overexpressing oncogenes, or you can potentially combine WEE1 inhibitors with chemotherapy or radiation that will increase S phase damage, or indeed, as we mentioned before, Olaparib combination that should also increase S phase damage. And then in addition, that damage can also lead to mitotic catastrophe if you override the G2M checkpoint, which WEE1 inhibitors can do because they're essentially activating aberrantly the CDK1 activity. So one of the other examples of synthetic lethality that's recently described is the effect of WEE1 inhibitors selectively killing cells that have lost the histone H3 mark, lysine 36 trimethylation. And in that scenario, cancers essentially have low levels of DNTP starvation, and WEE1 inhibition further leads to that DNTP starvation. This is a paper that came out last year from the Oxford group, Pfister et al., with Tim Humphries as the PI. And again, this is a very elegant example of how you can target a dependency of a cancer specifically because of the genetic deficiencies it has. And what the Humphries lab shown was that essentially WEE1 inhibition is synthetically lethal with the loss of histone H3 lysine 36 trimethylation, that ribonucleotide reductase M2 is the target of that synthetic lethal interaction. And really it's the genetic backgrounds where you have loss of the tumor suppressor gene SETD2, which is the methyltransferase, or indeed the overexpression of the oncogene KDM4A, which you just heard about from Johnathan, which is a demethylase, that leads to that increased sensitivity to the WEE1 inhibitor. So what we've been doing is we've been looking at the potential for WEE1 inhibitors to work as single agents in cancer cells that have higher levels of replication stress for example, those with cyclin E amplifications or SETD2 mutations. And we've also been looking at whether or not it's been possible to combine WEE1 inhibitors with the PARP inhibitor Olaparib. And in this particular example, which are data from Caroline Dive's lab in the Manchester Institute in the UK, it's a small cell lung cancer chemo-sensitive patient-derived explant model. So this is a model that's been generated from circulating tumor cells. And what we know about the genetics of this particular model is that it has the P53 and PRB mutation, which most small cell lung cancers will have, but it also has cyclin E amplification and also a PALB2 mutation, PALB2 being a regulator of BRCA2. And what you can see is that this particular cancer is very sensitive not only to single agent 1775, but also to Olaparib. So tumors regress even after only 21 days of treatment. What you then find is that those cancers tend to grow back once the treatment has been stopped. But what you can see in the bottom right quadrant is the combination of the WEE1 inhibitor with Olaparib and the effect that that has. So not only do you get all of those cancers responding very effectively and regressing, but in fact that regression lasts a significant amount of time. And in fact, out of the 7 animals that were treated, 5 of those animals were tumor-free for more than a year. So we're really excited about the possibility of combining DDR agents to specifically target replication stress. And in my last 2 slides, I'm just going to talk a little bit about the growth of DDR therapeutics. So this slide highlights just how much increased activity there's been in targeting DDR since 2005, where there are only 5 compounds in clinical development, 5 clinical manuscripts, to a situation last year where there were 34 clinical manuscripts describing 41 different DDR inhibitors. And again, you can see from a number of ASCO abstracts that have gone from just a handful in 2005 to more than 450 now in 2015. But I just want to end by saying that, when we're thinking about the targeting of those enzymes that are involved in chromatin modulation, what we'll generally find is that in addition to kinases, many of these enzymes are going to be more difficult to target. It's not going to be impossible, but it means that we really have to understand the mechanisms and the structural relationships of those inhibitors and how best to approach that. Because what you can see at the moment is that we've probably been targeting the low-hanging fruit. So even though 40% of the 400 or so DDR proteins that have been identified have enzymatic activity, about 80% of the current drug target focus is really on just those small number, 4% of the kinases within the DDR sort of pathways. And I think going forward, what we're going to be able to do is think about how we can identify inhibitors of these other enzyme classes, including those that regulate chromatin to great effect. And probably, again, it's going to be combinations where we see the most effect. So I'm going to stop there. And I just want to be able to acknowledge all of those people within AstraZeneca who've been involved in primarily the work I've talked about today on PARP and the WEE1, but also QDOS, where Olaparib was first discovered, and also those many different academic groups where really the great science has been done and where collaborations with drug companies really gives us an opportunity to understand the best way to use these inhibitors in order to be able to treat cancer. So I'll stop there. Thank you very much.
Thank you so much, Dr. Connor, and thank you to both of our speakers for the fantastic presentations and for sharing all their work with us. We're going to move on to questions submitted by our online viewers right now. Just a quick reminder, those watching live, you can still submit your questions by clicking the Ask a Question button below the slide window, typing the question into the message box, and then clicking OK. So the first question I'm going to put to both of you, and Dr. Whetstine, let's start with you. When considering tumor heterogeneity, what are some of the challenges that you've encountered in understanding tumor evolution and clonality, and particularly the response of tumors to treatment?
Well, I mean, as a field, it's become clear in the past, say, 5 years or so that as one is assessing, the emerging resistant clones or metastatic lesions, or even looking within the tumor itself as an intra-tumor, that there is this variability that occurs for both copies as well as mutational status. And it's very clear from a number of different groups that more than just us. We're looking at the mechanism drivers of these, but from the literature, it's very clear that this is not just a one tumor type problem. This is actually across many different tumor types, both solid and hematopoietic malignancy. So it's very clear that it is a problem. Some major questions are, how do you get such diverse changes? I mean, you just heard from Mark about various pathways or repair pathways that could influence the events. Alterations in those could result in some of the mutational events or some of the changes. We believe that in the context of copy heterogeneity, that the misregulation or the missignaling to epigenetics is probably driving a big portion. I wouldn't say all of it, but I believe that it's impacting a pretty big percentage of it. And through our most recent work in the lab that's unpublished, as we know that there are, this is not just a KDM4 effect, there's actually more to this than we appreciated before. So we think that there's many avenues to drive heterogeneity, but we believe that this epigenetic component is probably a big contributor.
Dr. O'Connor, any thoughts from you?
Yeah, and I think that tumor heterogeneity obviously is a key issue. But I think what's encouraging when we're thinking about targeting the DNA damage response is that, as I mentioned during the talk, we think that the loss of one or more DDR pathways is actually a very early event with the potential for it to be more homogeneous amongst tumor cells. And although it is possible to reactivate sort of point mutations, in many cases it won't be possible to reactivate that pathway. And so we think that the loss of DDR, and particularly in cancers that have high levels of genomic instability, that really represents an Achilles heel and something that we should be able to effectively target. So we are hoping that by targeting the loss of the DDR events is very early, and in particular targeting that greater level of dependency, we have an opportunity to really hit more of those cancer cells and have longer-term effects. And I think the fact that we have patients who, for example, been on Olaparib for more than 9 years now, and about 1 in 6 of the patients who've been on treatment for more than 5 years, actually argues that we can, if we identify, DDR loss events that are drivers of the cancer, have sustained impact and potential to help those patients.
Excellent. So you actually partially answered part of my next question, which was about whether DDR mutations are an early event. So the second part of that is, do you see bias towards the loss of certain DDR pathways in certain cancers, or does it seem to be more random?
No, I think there is a rationale. For example, in certain hematological cancers, for example, such as CLL, mantle cell lymphoma, the loss of ATM is an event that's associated with cyclin D amplification on chromosome 11q, 22, 23. So there, what you're having is a loss of ATM as a consequence of the amplification process of cyclin D. So that's a driver event that leads to around 25% of CLL having ATM deficiencies and about 40% of mantle cell lymphomas. In high-grade serous ovarian cancer, we're seeing up to 50% having losses in the homologous recombination repair pathway. So I think there will be differences, and I think that's why patient selection is going to be absolutely pivotal if we're going to be successful, because we have to know in any given scenario for a cancer that we want to treat, what is the DDR pathway or protein that's been lost, and what is the dependency that we can then target.
Excellent. So let me just, I'm going to stay with you, Dr. O'Connor, for another question. Is there a danger in the long-term use of something like Olaparib that genomic instability is being generated in normal or precancerous cells that might lead to the development of further cancers not associated with the original cancer?
So I think there has to be a finite increase in risk, given that you're increasing the level of unrepaired damage. But as I say, what we have been able to see now, at least for Olaparib in BRCA patients, we've had thousands of patients who've now been treated. The first patient that I showed you who responded was treated in 2006, but we've been treating patients since 2005, so it's 9 years that we've been able, well, in fact it's more than that, isn't it? It's 11 years now that we've been able to sort of follow those patients out. And what we have seen from that study 19, where we've been particularly looking to see whether or not there's increased risk of hematological cancer, is that there does not appear to be any statistically significant increase over and above what they get from the chemotherapy. So I think what we also have to do is put DDR-targeted therapies in context with chemotherapy. So we know that chemotherapy creates significantly more DNA damage. It's causing exogenous DNA damage. Whereas what we're trying to do by targeting DDR is actually kill cancer cells with endogenous damage, which is at a higher level than normal cells. So while I think there is a finite increase in the risk, we do believe that that risk is not the same as chemotherapy, and we think that the advantages outweigh the risks.
Dr. Whetstine, this is actually a related question that came in for you, and that is, do you think that chemotherapy could potentiate copy gain, not only resistance to chemotherapy, but also cause more genomic instability and a worse outcome than without chemotherapy?
So in that context, based on the data that we currently have and what we've been looking at is, we know that if we can drive copy gain events in certain regions, in the case of looking in breast cancer cells, by driving the amplification of the gene such as CKS1B, that we know that there's an associated reduction in response to, say, certain damaging agents, say platinum drugs. And so now the question is, and this is something that we're currently investigating, is the notion that if you sustain exposure to those types of drugs while this mechanism is being kind of pushed, will that promote you to potentially integrate or potentially inherit or pass forward those? And so instead of having transient events, you now can acquire cells that upon insult or genomic instability because of the therapeutics allow you to kind of get an altered version, which is a genetic change. We currently don't have an example of that right at the moment, but it may be too early in the sense of it may be that you need longer exposures, you may not find the right combination effects yet. But that's something that we're actually interrogating, but it is an interesting possibility because you can imagine that if you could remove it, if you could move from a case of a transient to a stable integrant, that you now would have a potential cell that has acquired, this additional benefit. So I think it's highly possible, and something that we're looking at right now.
Okay, I'm going to stay with you, Dr. Whetstine. This viewer says publications about genomic alterations in the tumor stroma have been contradictory. Do you have any personal experience in this area?
I don't have any experience in that space. What is interesting, though, and I will say this, is that in the context of being able to drive amplification, I mean, often if one looks at how people view altered DNA copy number, it's often thought to be a very potentially deleterious effect or pathologic in many ways. But we do know that your cells, your somatic cells, they can, different tissues can do this upon the right stimulation. With hypoxia, we're able to do it with all kinds of cell types. And we also show it's conserved in the fish. So what I would say is, maybe some of the contradictory information about copy number or alterations that one sees by looking at different tissues or looks at somatic conditions, some of this may be reflective of the conditions or the exposure or the growth factors or those types of things that are in there. And I think that, now that this work has begun to illustrate that there are, in fact, pathways and that there are mechanisms to control these things, I think having that knowledge and approaching that question that way may allow us to better understand some of these types of events that are occurring.
Dr. O'Connor, do you have anything you'd like to add about genetic instability in the tumor stroma?
So I think the microenvironment is clearly going to be important, and I think there are probably two aspects, one of which Johnathan just touched on, which is hypoxia. So there are questions about whether or not hypoxia can lead to the downregulation of certain DDR proteins, which again may provide opportunities for therapeutic intervention. So hypoxia has been linked, for example, to the downregulation of the homologous recombination repair factor genes RAF51. I think the other aspect is the inflammatory response. So again, a number of the agents, DDR agents, feed into NF-κB signaling. So again, when we're thinking about things like radiation response, it's going to be very interesting to see whether or not DDR inhibitors not only potentiate the damage to tumors, but also might protect cells in certain ways. The loss of ATM, for example, in the ATM knockout mouse, led to high levels of radiation sensitivity in most tissues of the body, but not, for example, in the brain. So neurons were actually radioprotected. And so, one aspect there that's very interesting that we're going to be looking at is the potential to use ATM inhibitors in combination with radiation in p53 mutant gliomas, where we hope to get impact on the cancer cells, but potentially protection there in that case, not so much necessarily through suppression of inflammatory response, but more about suppression of p53 dependent apoptosis.
Fantastic. Unfortunately, that brings us to the end of our webinar. We are regrettably out of time for this particular event, so it just remains for me to thank our speakers for the wonderful presentations, Dr. Johnathan Whetstine from Massachusetts General Hospital and Harvard Medical School, and Dr. Mark O'Connor from AstraZeneca. Please go to the URL now at the bottom of your slide viewer to learn more about resources related to today's discussion, and look out for more webinars from Science available at webinar.sciencemag.org. This particular webinar will be made available to view again as an on-demand presentation within about 48 hours from now. We are interested to know what you thought of this webinar. Send us an email at the address now up in your slide viewer, webinar@AAAS.org. Again, thank you so much to our panel and to Cell Signaling Technology for their kind sponsorship of today's educational seminar. Goodbye.